Thank you, and good morning. I am Jérôme Becquart, and I am Brainvectis CEO. At Brainvectis, in fact, we develop gene therapy products to treat neurodegenerative diseases, and our target is the brain cholesterol metabolism, which is often impaired in those diseases, in fact, downregulated in those diseases. So Brainvectis is a spin-off from INSERM. Nathalie Cartier is our scientific founder. She has a strong experience in gene therapy for the CNS, from the science to the clinic. And our preclinical director of preclinical research is Sandro Alves. So again, our target is cholesterol metabolism in the brain, which is often decreased. Our most advanced program is BV-CYP1 for Huntington disease, and our labs are located at CEA Medicen, and also soon at the ICM. So, to restore the brain cholesterol metabolism, in fact, our target is called CYP46. This is a key enzyme located downstream to the metabolism pathway in the brain, and it is specific to the brain. So we have shown that in several neurodegenerative diseases, this enzyme is decreased. So our therapeutic strategy to overexpress CYP46 in the specific disease brain area affected by the disease. So to do that, we use a very well-known and safe adeno-associated vector that code for the CYP46. And our most advanced program is BV-CYP1 for Huntington disease, for which we address the striatum, which, as you know, is the main area of the brain concerned in the pathology. Of course, the main advantage for this strategy is that gene therapy is a one-time treatment. So this is our pipeline. We are interested in neurodegenerative disease for which it has been demonstrated that there is an impairment of the brain cholesterol metabolism. So up to now, we have validated CYP46 as a relevant target in 5 different neurodegenerative diseases, and we have got altogether proof of concept in 8 different animal models of neurodegenerative disease. And to us, this achievement, to have 8 proof of concept, really reinforced, in fact, the validation of our approach. So for Huntington disease, our objective is to reach the clinic by 2020. For Alzheimer's disease, we are just starting evaluating new vectors that can cross the brain-blood barrier in order just to be able to inject intrathecally. In SCA, this is a spinocerebellar ataxia. In fact, we target the cerebellum, which is the main organ concerned by the disease. And we have demonstrated that, in fact, our approach could be relevant in 5 different types of SCA. And we are also interested in other 3 rare neurodegenerative diseases. So to develop our products, we have set up a network of partners listed here. You can see that it covers all the value chain from the mice models to the clinic, and we have been supported by the Satil de France Innov, with which we have a maturation program. So what are our needs? First, system biology methods. Firstly, to better understand the metabolism of cholesterol in neurons and in the brain. And secondly, to help us elucidate the mechanism of actions of our projects in the different diseases we are focusing. Then we need always new animal models. New vectors, as I told before, especially those that can cross the brain-blood barrier, so that we could be able just to think of intrathecal or even systemic administration. New injection technologies to better deliver the vector into the brain. And finally, biomarkers for the translational development, and clinical markers, of course, that are key to follow the disease progressions. Okay, with that, I'm finished.